Welcome to Meiosis I 360. Today, we are embarking on a journey into the most important biological process in the history of complex life. Without this process, every human on Earth would be a clone. There would be no evolution, no adaptation, and no you. We're talking about meiosis. While mitosis is a biological photocopier, meiosis is a biological shuffler. It is the process where a single diploid germ cell carrying 46 chromosomes undergoes 2 specialized rounds of division to produce 4 haploid gametes, each carrying only 23. This ensures that when a sperm meets an egg, the result is exactly 46, the magic number for human life. In this 20-minute deep dive, we will leave no molecule unexamined. Before the first division begins, the cell must prepare. This is interphase. G1 phase. The cell increases in size and produces the proteins needed for DNA replication. S phase, synthesis. This is where the magic starts. Every DNA molecule is unwound and copied. We move from 46 single-stranded chromosomes to 46 double-stranded chromosomes. G2 phase. The cell double-checks the DNA for errors. If a mistake is found here, the cell may undergo apoptosis, cell death, to prevent a mutation from passing on. The parts of the machinery. To understand the script of meiosis, you must know the actors. Centrosomes, the anchors that will pull the chromosomes apart. Microtubules, the fishing lines or spindle fibers. Kinetochores, the hooks located on the centromere where the fibers attach. Sister chromatids, identical twins of a chromosome. Homologous chromosomes, a pair of chromosomes, one from your father, one from your mother, that carry the same genes but different versions or alleles. This is the most critical phase. We are going to reduce the chromosome count from 46 to 23. Prophase 1, the 5 subphases. Most textbooks summarize this, but we are going deeper. Prophase 1 is divided into 5 distinct stages. Leptotene, the chromatin, condenses, the chromosomes look like thin threads. Zygotene, the synaptonemal complex forms. This is a protein zipper that physically binds homologous chromosomes together. This pairing is called synapsis, and it forms a tetrad. Pachytene, this is where crossing over occurs. The non-sister chromatids of the homologous pair break and exchange genetic material. Why this matters? This creates recombinant chromosomes. You are literally mixing your parents' DNA to create a code that has never existed before. Diplotene. The zipper, the synaptonemal complex, disappears. The chromosomes begin to pull apart but remain stuck at the chiasmata, the physical points where they swapped DNA. Diakinesis. The nuclear envelope fragments. The chromosomes are at their maximum thickness. Metaphase 1, independent assortment. The homologous pairs, the tetrads, line up at the center. But here's the secret. They line up randomly. The dad chromosome might be on the left for pair number 1, but on the right for pair number 2. This is independent assortment. The math? With 23 pairs, there are 2 to the 23rd over 8 million different ways they can line up. This is why you don't look exactly like your siblings. Anaphase 1 and Telophase 1. The spindle fibers pull the homologous pairs apart. Crucial distinction. In mitosis, we pull sister chromatids apart. In Anaphase 1 of meiosis, the sister chromatids stay together. We are separating the sets, not the twins. The cell then pinches, cytokinesis, resulting in 2 haploid cells. Between meiosis I and meiosis II, there is a short break called interkinesis. There is no S phase here. The DNA is not replicated again. This is the biological fail-safe that ensures the final cells only have 23 chromosomes. The centrosomes duplicate and the cell prepares for a second round of movement. Meiosis II is much faster. Think of it as a mechanical separation. We started Act 2 with 2 haploid cells. We will finish with 4. Prophase II. The chromosomes condense again. No crossing over happens here. Metaphase II. The chromosomes line up in a single file, not in pairs this time. Anaphase II. The centromeres finally break. The sister chromatids are pulled to opposite poles. Telophase II and Cytokinesis. New nuclear membranes form. The cells split. The result? You now have 4 daughter cells. Each is haploid. N equals 23, and each is genetically unique. Meiosis looks different depending on your biological sex. Spermatogenesis versus oogenesis. In males, meiosis produces 4 equal-sized functional sperm cells. This is a factory that never stops. In females, meiosis is asymmetric. During the divisions, one cell takes almost all the cytoplasm and nutrients to become a large oocyte or egg, while the others become tiny polar bodies that eventually wither away. This ensures the egg has enough energy to support a developing embryo. When meiosis goes wrong, nondisjunction. Bones break, and so does meiosis. Sometimes chromosomes fail to separate correctly. This is called nondisjunction. If it happens in meiosis I, all 4 gametes will be abnormal. If it happens in meiosis II, 2 gametes will be normal and 2 will be abnormal. This leads to conditions like trisomy 21, Down syndrome, where an individual has 3 copies of chromosome 21 instead of 2. We have covered the microscopic dance of the chromosomes, from the complex unzipping in Pachytene to the final separation in Anaphase II. Meiosis is a testament to the precision of life. It provides 2 main functions: maintenance of chromosome number. It prevents the genome from exploding in size every generation. And genetic diversity. It provides the raw material for natural selection to act upon. Every time meiosis occurs, it is a roll of the genetic dice. You are 1 in 70 trillion possibilities. Thank you for joining this Meiosis 360 Masterclass. Don't forget to like, subscribe, and keep exploring the wonders of biology.